Hi, welcome to our first podcast. We're going to be talking a lot about things, so I wanted to just get us started. Today we're going to be talking about evolution. So, what is evolution? We got a couple essential questions that we're going to be covering. First off, what is evolution? So, a lot of this is going to be a review from what you did in freshman bio. So, how does evolution work? What is the mechanism of evolution? So, how does natural selection work? So, that'll be the 5 variables, and you can think about things like directional selection with the moss. What are the different variations of selection? So, this will be getting into some additional details that you might not have covered, things like sexual selection. And lastly, in this podcast, we'll get the last bit for what is the evidence for evolution. So just since we're reviewing, might as well hit on the history of evolutionary theory. So the first person to kind of think of how traits are inherited in a population started Linnaeus, developed a system of classification based on what things look like, and that was followed by Lamarck. Lamarck is famous for his idea on giraffes, and that giraffes would be able to will themselves to change. They could want to extend their necks longer and therefore control their genetics. Now we know that that's not possible. You can't control your genetics. You can't determine what mutations or genetic combinations you have. But at the time, it seemed reasonable. If you wanted a longer neck, you could stretch and change it, and then those changes would then be passed on to the offspring. So what happened after Lamarck? We came to Darwin. So what is Darwin famous for? He's famous for coming up with the idea of natural selection, and he was able to do that in conjunction with other ideas from Mendel. So the connection between Darwin and Mendel was that there was a genetic component to it. So Mendel was able to then track genetics through using pea plants, and then that was something that we could select for and then pass things on. So what are the components of natural selection? There are 5 parts. The first part is that population growth has to be exponential. You have to have a lot of growth. When you have overpopulation, you can then select for something. From there, the population has to be stable in size. So if you have a lot of fluctuations, you can't really have natural selection going on. You have to be able to have the trait pass on from generation to generation. You have to have limited resources. That creates competition. Because there's variation, so 4 is variation, because there's variation, you can select for something. Different variants are then able to compete for resources and then able to reproduce. And because they're reproducing, you need to have that variation be heritable. So it doesn't really matter if I have some great mutation that allows me to get food faster. If I can't pass it on to my offspring, it's not going to be something that's selected for. So what is natural selection? It's the idea that certain individuals can have more kids and they can compete for resources. When you compete for resources, you pass on traits. So natural selection is the idea that you can pass on traits that are advantageous. So at the time natural selection was first proposed, it was seen as something really crazy, especially with the idea that Darwin was associated with monkeys, and that wasn't going to work out at all. So, but where's somewhere you can see it? Here's kind of the first part of evidence: artificial selection. How can it be that Great Danes and Shar-Peis are the same species? They can interbreed, they can have puppies. When you think of the great diversity in dog species, it really just shows how much you can select for traits. The same is true in other animals such as cats. So we can have desired traits. This is especially true, you can also see it in things like bananas and a lot of other plants that people have selected for through domestication. Artificial selection keeps going, so we can take different kind of subspecies or breeds or variants and make things like cockatoos, labradoodles, and that's another part where you're picking traits that you want. So poodles, right, this is artificial selection by humans. People wanted an animal that shed less fur. In some ways, people say that there's artificial selection going on right now with biotechnology and genetically modified organisms. Okay, so now let's get into some evidence for evolution. So one of the things that people always want to bring up when they're thinking about evolution is vestigial structures. If there's truly creationism, why would they create something that you have that has no purpose? So they're no longer used in an organism. For example, human appendixes. You can think of the tailbone. We have a coccyx that easily gets bruised, especially in figure skating. We don't use it anymore. It doesn't really have a use for us. But it did once upon a time. So ancestrally, the appendix was helpful for digesting bacteria. And people also relate vestigial structures to embryonic development. There are other types of structures. You might consider things like analogous structures. Here again, we're looking at root words. So vestigial, and we have analogous. Analogous structures are structures that have similar functions but are different. So an example would be a bird's wing, a bat wing, and an insect wing. They're all developed to fly, but they came from different ancestors. But they were selected for the same function, being able to have flight. So another vocabulary word that you might want to pick up on is convergent evolution. So similar selection pressures cause similar traits to evolve. Another example of this would be cactus. So in multiple parts, different deserts, they've evolved spines, really fleshy stems, and that's to conserve water. But they weren't originally related. Another type of structure is homologous structures. So going back to homo meaning the same. So we had vestigial, which meant you didn't need it anymore. We'd have analogous, meaning they were comparable, and we have homologous, meaning they arrived from the same thing. So the classic example is the arm appendage. So humans, cats, whales, and bats, they all have the same baseline of structures, and you can see they're highlighted here. And you can see they're highlighted here with the humerus, being the same bone, but different lengths and different functions in different animals. In addition, you'll see that they have a common ancestor. Another thing people use for evidence is biogeography. So once upon a time in the history of the Earth, all the land masses were together in something like Pangea. They'll look at where organisms are found, such as the sugar glider and the flying squirrel. They're found in the sugar glider and the flying squirrel. They're found in two different parts, North America and Australia, but they have similar characteristics. So what do? They think that they believe that they've evolved from one another, or they have similar environments. There's two parts to that, and that especially comes in handy when looking at cladograms and trying to look at relationships between organisms. So what happens besides natural selection? One of those mechanisms is genetic drift. So what is genetic drift? It's the idea that your gene pool events are random. So not only is nature acting, but so is the general environment. These can be things like earthquakes. Earthquakes can cause random events, but other examples include the bottleneck effect, founder effect, and geographic isolation, which we'll go into in more detail. The bottleneck effect is due to a random thing affecting the amount of genes in the population. So for example, the parent population had all blue and yellow at a certain ratio. When a bottleneck event happens, such as an earthquake, such as a flood, predators, all kinds of things, the number of certain traits changes, which could affect the population. So in the beginning, there were more yellow than blue. In the end, there's more blue than yellow. This happened a lot in cheetahs. So cheetahs have become so inbred that you can see a rapid shift in their genes. This has been traced back by DNA technology and then being able to compare the cheetahs. Another result that occurs from random chance is the founder effect. So this happens a lot when you have new islands. So a few individuals become isolated. So on an island, random animals can come on there through sticks, through plants, through the wind. They happen by chance. You don't get selected to become a founder. The new population is not the same as the original. It may be one individual out of 100 that represents this new population. So an example of the founder effect could be blood type. So once upon a time, going across the land bridge, you'll see that we have a majority, 80 to 90%, have the O blood type in Northern America, and it increases as you go south because an individual who had the O blood type founded these new areas and was then able to spread it on to the entire population. Another example are certain syndromes and diseases. This is especially true for areas that are isolated up in the mountains. Geographic isolation is related to founder effect. If you're geographically isolated, you're less likely to have gene exchange with other areas. So when a physical separation or isolation results in a new species. So When Darwin went to the islands, they were physically separated, and it created new species. So this happens a lot of times when you have a new area that an animal needs to found, and they need to niche. So this is part of divergent evolution, which is also known as adaptive radiation, so many species evolving from one ancestor. So if you look over here at all the bird beaks, these all came from one original founding bird that went to the islands. But because of different needs for nuts and plants, they resulted in different types of beaks. Here's another example of geographic isolation, where species were once the same, but over time, because they were isolated, have changed. This tends to happen a lot with river separation or mountain ranges. Gene flow is the basis behind all of this. If you're genetically isolated, then you don't have any genes flowing back and forth. But you can have migration, which allows genes to flow. So for example, the human population today has a lot of gene flow due to things like modern technology that allow flying, boating, swimming, which allow genes to intersperse. This changes allele frequencies. Instead of there being certain frequencies set in one area that was limited by mountain ranges, now alleles can be found all over the place, making it harder to track genes. Next up is another vocabulary word, phenotypic polymorphism. This basically says, if you break it down, phenotype is what you look like, poly meaning many, morph meaning traits. So you have many, a variety of physical phenotype traits or morphs. This is caused by variation, genetic polymorphisms, which is something that happens at the DNA level that we'll hit in the second semester, and variation that's not inheritable. So this is usually due to environmental factors. Another concept related to evolution is the idea of fitness. So a lot of times people think of fitness as working out and getting big muscles and being able to run cardio. In the evolutionary sense, fitness is all about having kids. So fitness is being able to have traits in the gene pool, and relative fitness is compared to others the same age or ability. So fitness is one means you're contributing all. So this tends to happen a lot with clones. If you're a clone, all of your genes get passed on. Generally, in sexually reproducing animals such as humans, 50% is what gets passed on. 0 means you don't have any kids. I don't have any kids, so right now I have 0 fitness. So I'm not very evolutionarily quote-unquote fit, even though I try to work out and go running. But in evolutionary sense, I'm not at all. So we've covered so far some of the mechanisms of how selection works. Now we're going to hit on some of the specifics. So not only was there natural selection, which was typically the environment chooses, but there's also different types within that. So there's stabilizing selection, which is the idea that the average is what gets maintained. So this tends to happen a lot when, if you're too big, it becomes an issue. Too small, it becomes an issue. So stabilizing selection tends to maintain the norm, keep the status quo. Disruptive selection says that there's two extremes, and the one in the middle is the problem. So the darker colored snail is better for the dark environments. The lighter colored snail is great for blending in or maybe looking like bird scat. So you have disruptive selection. The two extremes are what matters, and over time the one in the middle is eliminated. This is really common for speciation. There's also directional selection, where one phenotype is selected. So you're moving toward one end. So this is the famous peppered moths experiment, or case study actually, that happened in Great Britain with a lot of the Industrial Revolution, where there was selection for the black moth, and as time changed and pollution left, the directional selection went back toward the white moth. So when does this stuff start getting tricky? With humans. So a lot of times when we're thinking of disease, you think, okay, things just get wiped out, survival of the fittest, everything's great. That's not always true with things like heterozygote advantage. So the classic example is malaria. So in malaria and sickle cell trait, when you have both recessive alleles, you'd think that sickle cell trait, which is pretty bad, it means you have a lot of difficulty breathing, it's hard to get upstairs, you have issues with blood clotting, you think that would be slowly eliminated. All the people with sickle cell trait would die off. However, people who are heterozygotes, meaning they have regular blood cells and sickle cell traits, at the same time and that's simultaneously expressed, such as seen in this blood smear. So to continue on, to finish up the idea of heterozygote advantage, it's that the heterozygote has greater fitness than just the dominant by itself or the recessive by itself. So the heterozygote then continues to be selected for despite recessive alleles not being favorable. So the perfect example again, sickle cell anemia, where the heterozygotes are resistant to malaria. And there's also sexual selection. So it's the idea that males and females look different due to their different investment in their gametes. So males don't have to invest in a lot in their gametes, so they tend to have to then compete for females, while females are more choosy. This competition can be within the gender and across the gender if we're trying to be able to have offspring. So this again has to do with higher fitness. So why does the male peacock look like the way he does? He's got to compete with other males to convince the females to have sexual relations so that he could pass on his genes to his offspring. So those were all good points, but there's a couple constraints on natural selection, and I wanted to finish up and conclude with those. So populations are not perfectly suited because the environment's constantly changing. As soon as you think you're ready, the environment changes. This is the idea behind the Red Queen hypothesis, that for Alice in Wonderland, she always had to be running just to keep up. What else is there? There's also that evolution's slow. So if you think about a human lifetimes live 80 years. It's pretty hard for evolution to happen and have selection within humans just because we live for such a long time. While you can see evolution on a quicker scale with things like bacteria, because their reproduction is something more like 2 hours, which is a lot quicker than humans. There's also that adaptations are compromised. So you don't always have a perfect fit. You kind of tend to have something that works best in a certain situation. Ideally, you would, ideally creationism, if it was true, you would get something that would fit perfectly to that environment. But that's not what happens. What happens is you have the best that you can do given the time, given the environment. Natural selection can only pick it, and it can't create the variations. This is where a lot of times where mutation comes in, and we'll cover this a lot when we get into gene expression. But while I was saying before, you can't pick it out, it has to come from within the population. So for example, if we look in our class, we only have certain variations. We can't make somebody with wings because it's not there to select for. We can't pick a person who's immune to AIDS. It has to show up first naturally for it to happen. So that was the end of the slideshow, and I wanted to thank you for spending time with me, but I also wanted to make sure I spent some time to summarize. So to summarize, in conclusion, we found that evolution was the change of populations over time, and it was affected by many thinkers, including Mendel, who found the genetic component, and Darwin, who was able to put it into words using his evidence from the Galapagos Islands. We were able to look at the mechanisms, looking at natural selection and its variations, and things like sexual selection. And we were also to look at other mechanisms, such as the founder effect, where we saw genetic drift, where evolution could be, quote-unquote, random and not just be all about natural selection itself. We also looked at the constraints of natural selection and why it wouldn't always create the best possible outcomes for the best possible environments. So hopefully using that, you can answer your questions from the very beginning of the lecture. And take care. I'll see you next time.